Now let's take a look at microcytic versus macrocytic anemia. And these are diagnosing anemias based on red blood cell size. The first type of red blood cell disorder, size disorder, let's take a look at, is the normocytic, normochromic. This typically means that normocytic means that the size of the red blood cell is normal, and the hemoglobin content is also normal. So under these conditions of anemia, there is no inherent problem with red blood cell development. So red cell development is normal. These types of anemia come from blood loss through injury, hemolysis due to disease, or infections. Now, if we look at a change in red blood cell size, so let's look at the microcytic anemia. Whenever it's microcytic, it is going to be hypochromic. So under these conditions, the red blood cell is going to be, the size is going to be decreased. They're going to be smaller. The total hemoglobin content is also going to be decreased, of course, because smaller cell will hold less total hemoglobin. The hemoglobin concentration will be normal unless there's a thalassemia. Now let's compare this and contrast with macrocytic anemia. Macrocytic anemia is going to be hyperchromic. Under these conditions, the red blood cell size is going to be increased, the cell size is going to be larger, and naturally the total hemoglobin content is also going to be increased. Now, what generates these two particular types of anemias? With microcytic hypochromic, this is largely due to either an iron deficiency, or in the case of a genetic error, a thalassemia, a genetic error in the alpha or beta globin genes. For macrocytic anemia, the primary causes of this are a vitamin B12 deficiency or a folate deficiency. Now, how do these causes rectify with these size differences in red blood cells? For macrocytic anemia, these cells have defective cell division. So during the time of hematopoiesis, when the red cells have divided, are dividing before they extrude their nuclei, they are defective in cell division, so cell division is essentially slowed. And this is because of the deficiency in vitamin B12 and or folate deficiency. These two factors are involved in one carbon metabolism. The most important aspect of this is we have a thymidine deficiency. Remember the T in DNA replication. If we have a deficiency in thymidine production, we'll have a deficiency in, or slowing, of DNA replication and a slowing of cell division. This will lead to unusually large red blood cells. For microcytic anemia, under this case, we are not properly maturing the red blood cells. So that under normal conditions they have sufficient iron so that they have their synthesis of the hemoglobin, and then they iron load the heme bound in the hemoglobin, and they arrive at a proper size. Think of these as they are just waiting and they continue to become smaller, so they're not released into the bloodstream after synthesis during erythropoiesis, and the cells are smaller. So that's not properly maturing. This is due to iron deficiency because they are not maturing at a proper rate without the iron. Now these two types of anemia are typically treated with dietary iron. So dietary iron as a supplement. Remember that vitamin C enhances uptake of dietary iron because it serves as part of the redox reaction in order to convert the ferric to ferrous state. For macrocytic anemia, we are talking about typically a folate supplement in the diet, and very often, because these two biochemical pathways are related, a vitamin B12 supplement also. One other portion of the blood test that will also show the macrocytic and microcytic, but it will not differentiate which one is which, is the red cell distribution width. If we have normocytic cells, that means that the cells have all approximately the same size. So their red cell distribution width is going to be very narrow. If this is cell number and this is cell size. With microcytic and macrocytic anemias, we will have some normal cells. And under these conditions, we will have the normal cells that will band in the normal cell size, but we will also have either small or large cells included in there. So the width of cell size becomes wider, hence an increase in red cell distribution width.